Hello everyone. So in this presentation we will discuss about plant disease forecasting and why forecasting is done, why it is important, and what are the different factors using which forecasting is being done, and some of the examples of plant disease forecasting, and also the use of computers in plant disease forecasting. All of these points will be discussed in the coming slides. So first of all, definition. It is prediction for the appearance of disease in a particular area before its spread to prevent yield loss. For example, sometimes farmers can request the authorities for getting forecast about a certain disease, or in some cases scientists or universities working on crops or some other authorities can do forecast for a particular disease which appears again and again in a specific crop. So they can do forecast for that in that particular area where the disease spreads the most, and they can tell the farmers in advance so that they can apply the required control measures so that the yield losses caused to farmers can be decreased. And the disease forecasting consumes a lot of time because a lot of data needs to be collected for a forecast. Number of resources are also used, so large amount of money is also invested for a particular disease forecast. And but the farmers can be informed in advance about a particular disease, and control measures can be adopted on time. Then here, information required for a particular disease forecast. So there are three factors which affect the disease forecasting. If the data related to all these three factors is accurate, then we can get a good disease forecast. So these are host plant factors, means all the factors related to the host plant, the crop plant. Pathogen factors, these are the factors related to the disease causing organism, the microorganism. And the last one is environmental factors, the climatic conditions, the weather conditions of that area for which the disease forecast is being done. So the information related to all these factors is required if we want to do a disease forecast. Firstly, host factors. So there are a number of host factors which affect disease forecast, but three are listed over here. So first one is presence frequency of susceptible host in that area, means whether that crop is cultivated again and again every year, means what type of cropping system is followed over there, monocropping or they diversify the crops. If they cultivate one crop again and again, then it is most likely that the disease will appear. So what is the prevalence of that susceptible host, means the plant in which that disease is caused in that area for which we are doing the disease forecast. So when we do a disease forecast, then we do it for a particular area. Then second one is reaction of host during its growth and development in response to pathogen activities, means how the host plant reacts or responds to the different activities of the pathogen during its different life stages. Means some diseases affect the plants at seedling stage, for example damping off. Others affect the plant at when the plant is grown up, for example rust in wheat. Means the diseases affect plants at different stages. So what is the response of the host plant? And the last one is number of susceptible plants and their distribution. So what is the population of the plants in which the disease can be caused? Sometimes the spores scatter to different locations, to the remote areas, and then they travel back when the crop season is again there. So what is the number of susceptible hosts? What is their distribution? Means if the crops are cultivated in other nearby areas, or they are cultivated at far areas, so if the population is more, the crop density of that particular species is more, then it will spread the disease very easily. But if the crops are scattered, means a crop is cultivated, then the same variety is cultivated at a large distance, then there will be less chance of the disease spread. And pathogenic factors, all the factors which are related with the pathogen. So first one is the different stages of the disease cycle of the pathogen. Then amount of the primary inoculum, when the disease is spreading, what is the amount of primary inoculum, then the secondary inoculum will also be in it. Then dissemination of the inoculum, whether the inoculum is being disseminated, it has traveled using vectors, whether sufficient amount of vectors are present to transmit the disease, or there is less amount of vectors, or there are no vectors which spread that disease. Then germination of the spores, then the spores are getting favorable conditions for the germination or not. Infection and incubation period, then infection caused by them, then their incubation period is less or it is more, means the multiplication of some pathogens takes longer time than others, some take very short time. Then sporulation of the pathogen on infected plants, the spread of the spores, re-spreading of the spores, this can also be called as secondary inoculum, when they spread again after multiplying from one plant, like from infected plants to the other healthy plants present in that field. Then potential of the inoculum, it is very important point, because sometimes inoculum is present in large amount but its potential is very low, then the chances of disease will be reduced. But if the spores are present even in small amount but their potential, their strength of causing disease is very high, then the disease will widespread. The last one is presence of number of pathogens in soil, water, seed or other planting materials. Like for planting we use cuttings, we use budding, layering. Those plant parts can also carry disease into the healthy plants. Whether those pathogens persist in soil, whether their spores rest in the soil, in water or in seed. For example, a number of fungal diseases are mostly seed borne. And the last one is environmental factors. What are the weather conditions of that area for which we are doing disease forecast? So major factor is humidity or moisture. Most of the fungal microbes grow at a faster rate when the moisture conditions are there, means the relative humidity is more than 80 or 90% for most of the diseases. And then is temperature. Temperature is favorable for that pathogen or not. Basically if the temperature remains low, then it can cause the spread of fungal diseases. Means what are the temperature conditions in that area. Also other factors like light intensity will also affect, wind velocity will also affect, means these can cause the spread of the disease. So, and the last one is weather conditions during the season of crop and intercrop periods. The season of crop means the time for which our crop is in the field. Then intercrop period, the time lapse between the two subsequent crops, means we have harvested one crop, then the field is free for some days and intercultural operations like tillage is being carried out for the next crop, means that time when the field is free, means the time span between two different crops, between harvesting of our first crop and up to the sowing of our next crop, that is called as intercrop period. So what were the weather conditions in that time? Conditions for practical gains for forecasting, means if we are planning to do forecast for a disease, whether it will be useful for us or not. Practical gains means whether we will gain something positive from the disease forecast or not. So here I will explain. So first of all, no, it is not done for all the crops. We cannot do forecasting for the crops which are sold at very low price or which are cultivated on small scale. They are not basically important. So we only do forecasting for the economically important crops, the crops which are cultivated on large scale in different countries or in a particular country, almost in all the states or in a number of states. Next is for major disease. The specific diseases of crops. For example, wheat, rust, then smut in wheat, blast in rice. These are the major diseases of particular crops. They affect the crop again and again. So major diseases, these are the diseases which have ability to cause an epidemic. They can spread in a very short period of time, and it is always the chances are higher that disease will spread. So for major diseases. Then in case of pathogens showing mutation or changes in behavior, means a pathogen which changes its behavior and in a very short period of time. For those also we do the forecast. And then when management practices are known for a disease, weather forecasting will only result positive if we have the management practices, the control measures of that particular disease. If the control measures are not known, then there is no use of the forecast. Means we cannot do anything even if we have the forecast, but if we don't know how to control that disease. So we should be well aware about the control methods for that particular disease. The last one, when there is complete knowledge related to weather conditions in relation to disease. For example, if a farmer wants to do a forecast or plant scientists want to do a forecast, then they should compile the data properly. They should have full record related to the temperature conditions, day temperature, night temperature, rainfall. We should have complete knowledge related to the weather conditions. Also in relation to the disease, means what are the conditions in which the disease prevails, what are the favorable conditions for the spread of a particular disease. Then methods of the forecasting. So there are basically 4 methods which are used for disease forecasting. First one is based on primary or initial inoculum. Second one is based on the weather conditions, then based on correlative information, and last one is computer based disease forecasting. So first one, based on primary inoculum. So here are the points mentioned. So presence of initial inoculum. If the initial inoculum is present, then there are chances that disease will spread. So means we compile this whole data and then we do a forecast. Then viability and density of the inoculum in soil and air and propagating materials. So whether, for example, initial inoculum is present, so we should know whether that is viable or not, means that has ability to cause a disease or not. Then density, the population. Then testing for presence of pathogens. We can do different types of tests to know whether the pathogens are present. So in air, in soil. So for testing them in air, we use different air trap devices, and in soil monoculture method is used. And then planting materials are also tested. For example, if we are using cutting or buds, then those materials should also be tested for the presence of the spores or for the presence of that pathogen. Then testing after taking random samples. We take random samples from the area where the disease exists, and then tests are done. And the last one is information is recorded and telecasted to farmers. So based on this whole data, the scientists analyze for disease forecast whether there will be the spread of disease or not. Then the whole information is telecasted to the farmers by different means. Second one is based on weather conditions. So these are the 4 factors. So first one is temperature, relative humidity, rainfall. Conditions are also tested at soil level. So what was the temperature? So we should know whether these all conditions are favorable for that disease or not. So what was the temperature range in that area? What is relative humidity over there? What is rainfall? means the amount of rainfall in that area, and the weather conditions at soil level are also tested, means night temperature, humidity in the soil or humidity near the root area of the plant, etc. And based on correlative information, this method includes the use of data collection. For example, if we collect data each year for a particular crop and for a particular disease, means what were the conditions when the disease was caused and the disease spread. If we know all the conditions which prevailed during that time, means if we know all the temperature, humidity, rainfall related conditions and many other factors related to that, if we have complete knowledge, data is recorded each year, then we can get to know in the future time also that whether the disease will prevail or not, because we will be familiar with all the conditions in which the disease existed. So the past data and the present data are compared and the probability is tested that whether there will be a disease or not. So this is correlative information by relating the data of past and present. And the last one is computer based disease forecasting. So this system is basically used in the countries which are economically very well developed countries, or the countries having advanced technologies, and this system is very useful. It is easy, it consumes less time, means once they have developed the software or program for the forecasting of a particular disease, then they only feed the data provided by the farmers and they give them the forecast for the disease. So one of the example is Blightcast. It is used in USA. It is for the potato blight, and if American farmers require the forecast for that particular disease, then they contact the Blightcast and they ask the farmers for the data needed, which is discussed in the next slide. So the farmer needs to record data related to the maximum temperature, minimum temperature, humidity and rainfall. If they require the forecast, then they give that data to the Blightcast and the operators feed that data into the computer or into that software, and the results are generated in seconds, and within minutes the farmers are informed about the disease forecast, whether there will be a disease or not, and they also suggest them with the suitable management practices or the control methods which they can use for the control of that disease. So this is the data which is required for computer based forecasting. So minimum and maximum temperature on that day. If a farmer is asking for a disease forecast from a particular agency, then they will generally ask that what was the maximum temperature, what was the minimum temperature, what was the temperature during day, and also the temperature during night. And second one is relative humidity during different hours, means during different period of time on that day what was the relative humidity, maximum temperature at which relative humidity was more than 90%, also the minimum temperature at which relative humidity was more than 90%. So they basically want to know what was the temperature range in which the relative humidity remains more than 90%, the low temperature at which it remains 90%, and also the higher range of the temperature at which it remains more than 90%. And also the data related to rainfall for the past 24 hours is required for the disease forecast. And these are some examples of disease forecasting, means these are the diseases for which disease forecasting can be done. So late blight of potato, so for late blight of potato some Dutch rules are applied. According to them, they know the data related to the temperature, the minimum temperature, the night temperature and the maximum temperature, and also the weather conditions. The weather conditions need to be cloudy, and also the rainfall conditions. And then for blast of rice also they give that it requires minimum temperature at night. between 20 degree to 26 degree centigrade, and also relative humidity more than 90%, then there are chances that the blast of flies will prevail. And also for wheat rust, the amount of primary inoculum, there should be large amount of primary inoculum present to spread the disease in distant areas and also in the nearby areas. So these are some important points related to plant disease forecasting, and I hope you find these useful. Thank you.